Hi, today this video we see about proteomics using Expasy tools. What is proteomics? Proteomics means it's a large-scale study of proteins and the structure and physiological role or functions. So we will study the proteins by using Expasy tools. The term proteomics first appeared in 1997. So using this proteomics, we can able to investigate protein expression patterns at given time in response to a specific stimulus, but also to determine functional protein networks that exist at the level of the cell, tissue, or whole organisms. And after we study the rates of protein production, degradation, and steady-state appearance, how proteins are modified, that is, we can able to study post-transition modifications such as phosphorylation, etc. The movement of proteins within subcellular compartment also studied using this proteomics. The involvement of proteins in metabolic pathways. So we can able to see how the proteins will be involved, converted in the metabolic pathways. It also easily studied through this proteomics in our tools. How proteins interact with one another. That also we can able to easily study through, understand through the proteomics. By using this bioinformatics of studying proteins, the tools will easily used to identify the functions or any properties of the proteins, and all this investigations are being done through the proteomic tools. So let's see about what is Expasy. Expasy is an online bioinformatics resource operated by this SIT, Swiss Institute of Bioinformatics. It is an extensible and integrative portal which provides access to over 160 databases and software tools and supports a range of life science and clinical research areas such as genomics, proteomics, and structural biology to evolution, biogeny, and systems biology and medical chemistry. The individual resources, that is databases, web-based, and downloadable software tools are hosted in a decentralized way by different groups of the SIT, Swiss Institute of Bioinformatics, and partner institutes. This was first created in August 1993. Originally, it was called Expasy, means Expert Protein Analysis System. So Expasy means it's an Expert Protein Analysis System and acted as a proteomic server to analyze protein sequences and structures and two-dimensional gel electrophoresis. So we can be able to analyze primary, secondary, tertiary structure all through Expasy tools. So common procedures for how to use this Expasy tool means go to the Expasy. It will lead to the Expasy homepage. We have to type what are the tools we know we need. Then when you have to search the tools and find the tools, and then you have to click the tool. It will take you to the homepage of the proteomic tools. Here they have one small box. Then you have to paste your protein sequence, interested protein sequence or accession number. Then you can click compute parameters. Sometimes in some tools they may be shows the perform. So you paste the sequence and you click perform. Or otherwise, sometimes you have to translate the sequence. The small box will be there. That is after pasting the sequence, you have to click translator. So this is the common format for procedure. So first we see about the physiochemical properties of protein sequence. So the tool used for studying analyzing the physiochemical properties of our protein sequence, we have to choose a tool is Protoform tool. So protein parameter is shortly referred as the Protoform tool. By using this tool, we can be able to compute the various physical chemical parameters for a given protein stored in SwissProt or TRMBL or for user-oriented protein sequence. That is, we can able to analyze or compute the parameters. The sequence maybe we can able to retrieve from the SwissProt or TRMBL or our own that is our synthesis the protein sequence. Also, we can able to evaluate it by using this protoform tool. So, in protoform tool, we can able to find out the molecular weight, theoretical isotopic point, amino acid composition, atomic composition, extinction coefficient, estimated half-life, instability index, aliphatic index, and grand average of hydrophobicity. This all we can able to study in the or see analyzed in the protoform tool. So it's for the procedure. We can go to Google search, type Expasy, and then enter click Expasy, go to Expasy page and enter specific proteomic tool. Or otherwise, we can go to the Google search and then type as the protoform. It will directly to the, when you click the protoform, it will get to the protoform tools from page. So here we have to paste the sequence, our industrial protein sequence, and click the compute parameters. Then we get the results of all the parameters of our protein sequence. This is one of the proteomic tools. Second one is we can be able to find out the peptide mass fingerprint of protein sequence. That is, peptide mass fingerprinting is also known as protein fingerprinting. It is an analytical technique for protein identification in which the unknown protein of interest is first cleaved into smaller peptides whose absolute mass can be accurately measured with mass spectrometers such as Monditoph or ESR-TOF. So these are the way we were able to identify the peptide mass. So peptide mass means it cleaves the protein sequence. The protein sequence maybe we can detect from the Sysprot or our user-entered protein sequence. Then we have to cleave this protein sequence with our chosen enzyme and compute the masses of the generated peptide. The tool also returns theoretical isotopic point and mass values of the protein of interest. If desired, peptide mass can return the mass of peptides known to carry post-translation modifications. So this is also we can able to that is we can search through the peptide mass. Here also we have to paste the sequence and then we have to choose the enzyme, which type of enzyme can be used for cleaving this protein sequence. So enzyme means trypsin, chymotrypsin. So you already know each enzyme have its own cleavage site. So select this enzyme and then you have to perform. So from this we can be able to find out the how many peptides are cleaved and then which peptide having a high molecular weight or which peptide have a low molecular weight. This will be seen in the peptide mass fingerprint. Next we see about peptide cutter. It is also similar to that of the peptide mass fingerprinting. So it's the same one. We have to choose the protein enzymes. And another one tool is Peptra. So what is Peptra means? We got to put the just we have submit the protein sequence. It will draw the peptides. So we have to give a primary sequence. It will form linkages with the protein that given protein sequence. It form a structure, peptide structure image. That tool is Peptra. Then next one is Proteus. That is the combined transmembrane topology and single peptide predictor. So here also we have to paste the sequence and then we can able to find out the transmembrane regions. Next one is Translator. So in this tool we have to give a DNA sequence. It will translate it into the protein sequence or RNA sequence. It will that. Then we can able to find out the by using this translation tool, we can form in the DNA sequence summation. It will contain the protein sequence. So from this, we can find out the open reading frames or mostly in the red form, and then it will helpful for the selecting for the initiator of the amino acid sequence. So this is about the translation tool. And next one is deduction and alignment of repeats in protein sequence. So we have to find out the repeat of sequence repeats in the protein sequence can be studied by using the tool radar. You have to detect and alignment of repeats in protein sequence can be detected by using the tool radar. So for the radar sequence, we have to submit it. We can get the how many, what are the sequences having a repeated sequence, where it is the sequence will be repeated, will be seen in the result. Next is the secondary structure prediction. That is, we can use the SOFAMA. SOFAMA, or another one is another one type of secondary structure prediction. That is GOR, G-O-R, GOR. It is also we are used tool. Another one tool is for the secondary structure prediction is CFSP and Choseman House, and this is also used. So there are three different type of secondary structure tools will available. Third one, so secondary structures are the beta helix, alpha helix, the random coil. So in this sequence, we can be able to know what are the secondary structures may be present, and then which type of secondary structure is predominant in the given sequence that should be studied or analyzed. So other than that, secondary structure, you can be able to see in transmembrane regions. So for this, we have to use the tool is TEM-PREDICT, TM-PREDICT, that is transmembrane prediction. So we know the transmembrane, that is the protein will be present inside the lipid layer, another in the first layer, that is called the transmembrane regions. So these are the tools we have studied in this seen in the video. So next we go to the first say about proteomics tool. First one, Protoform. So already told we have to type Expasy. So when you have to type Expasy, the tools also come that translate Protoform, or we can use Expasy tools. So we directly go for Protoform. So it's from a Protoform to Expasy. So this is the page for page of Protoform. So we know what is Protoform means. It's a protein parameter. It's a tool. Already I explained it is a tool which is computing various physical chemical parameters for a given protein stored in the Sysplot or TRMBL or used to enter protein sequence. So using this tool, we can be able to compute or analyze these parameters. So here we have to place the sequence. So here we have to place the protein sequence or accession number. So we have to take what we do for the protein sequence. We have to retrieve protein sequence from NCBI or Sysplot. So already I taken the retrieve the protein sequence from NCBI, that is GenBank. So this is the protein is collagen from the animal Mus musculus. It's a partial substitution of collagen. So here I copied the sequence and paste it here, and just you have to click compute parameters. Wait for a few minutes. So in this already we know I told that is what are the parameters we can be able to compute, that is amino acids, molecular weight, isoelectric point, and composition of amino acids. So here a total, so it is very typical to identify the amino acid composition in a large size of protein molecule, and also find out the negative charge amino acid or positive charge amino acid composition. But the tool, the bioinformatics tools, is very helpful for the scientist or easily find out the and compute the physical properties of the one further gene of interest. Protein of interest. So atomic composition, that is carbon, how much carbon will be present, then formula, total number of atoms, and then half-life period, and the N-terminal amino acids of the sequence, its half-life period and stability index. The protein may be a stable one or unstable one. Aliphatic index, the protein may be a hydrophobic or hydrophilic. That's also be present in the tools. So this is about the proteomics tools or Protoform. Next we see about the second tool, what we see, that is peptide. peptide mass. So or otherwise we can go for the Expasy tools. So this is switching. So we are find out here large tools will be present. So we are going to search. We can type it as peptide mass or peptide. There are a lot of tools will be here. Peptide mass or otherwise peptide cutter. We can able to use or find peptide. These are the we can use for the cleaving the protein sequence. So first we see about the peptide mass. So here he gives about the what is peptide mass. Then once again here we can once again paste the sequence. So already told that which enzyme. So how we get the peptides. So we have to choose that is the system in the protein sequence will be in the reduced form or non-reduced form, or it will be treated with acrylamide, or it is or methionine will be oxidized form, or then enzymes. There are different more enzymes will be there, that is trypsin, and then higher specificity, and citrin or cyanogen bromide, and then cleaved only at lysine position, at C terminal only we cleave, or otherwise N terminal lysine will be cleaved, or likewise arginine, aspartate, or combination of amino acids. Then chymotrypsin is not before the B. These are the one letter code of amino acids. Then trypsin, chymotrypsin, and the trypsin. So any enzymes we can have to use. Using this enzyme, you can cleave how many peptides will be seen, and along that each peptide have a molecular weight also found in the peptide mass. So now we choose trypsin. Then just we go to fed form. Click the fed form. So already we seen that is having a amino acids is 1140. So always you have to check how many base pairs in the protein sequence in the starting stage only. If you have to copy the sequence at the time any one of the amino acids will be missed in the starting or in the end, the result will be wrong. So always check how many amino acids we have chosen, that amino acids all are present in the pasted submitted tool. Then here we are using enzyme trypsin. Here it is cysteines are in a reduced form, methionines have not been oxidized. So in this all the peptide mass cleavages, small molecular weight peptides are not being seen. So here its isotopic pointer 6.11. It's a mass molecular weight average masses. This one how much? So always protein unit is Dalton. So here how many peptide sequences are there? So 1140. So there are large number of sequences there. Peptides are there. So this one is the high molecular weight. The position cleave between the protein sequences 931 amino acid to 980 amino acids. So second one, the last one, only 4 amino acid, that is 927 to 930 position. So here mostly 92.9% of the sequence will be covered. So remaining peptides are in less than 500 Dalton. So they are will be neglected. So this is the one we can have to leave a list of masses for printout. Only the mass will be seen here. So this is about the peptide mass. So first we have to try it at trypsin. Then we can go for the backup. We have to choose chymotrypsin. Then once again perform. So it's molecular. Previously trypsin we see in the position is high molecular weight is 600, I think 900 system something. But here it is 138 to 263. Position having the high molecular weight. So last is the fourth one, that is here 927 to 930, 927 to 931. Position is present in the trypsin. Here it is chymotrypsin. The position will be deferred. So here also the number of peptide will be deferred between different enzyme cleavages. So next So peptide cutter also similar one. So you have to place the sequence. So here we can use some chemicals also. Here are some microbial enzymes also present in the peptide cutter. So we have to choose all available enzymes at the point. So this will give at the same time it clears all the using all the enzymes or chemicals. It gives the results of the protein. So you have to check it is all available enzymes and chemicals. Or otherwise, if you want only one, also we can select it and then we perform. So we have to perform 1040 amino acids. So the results for the in peptide mass we go to count how many cleavages are there. But here it is directly given the amino acid, that is arginine C protein is 60 cleavages. Likewise, chymotrypsin, pepsin 129 at the pH of 1.3. So if we have to done it in the lab, it will take more time for the find out the peptide. So first you have to use a cleavage, then you have to analyze this page, then you find out the molecular weight of the peptides. But it will take more time. But this it is the bioinformatics tools of proteomics will help us at a single time within a 5 minutes or a few seconds we can run with the how many enzymes 1, 2, 3, 4, 5, 6 enzymes, not only chemicals also formic acid more than I think 20 or 15 enzymes and the chemicals will be run act on the one protein. It gives the different type of cleavages. So these are the different position where the cleavages will occur. So here we cannot be able to find out the mass. We can able to only see in the number of cleavages and where the cleavages will occur at the position. That will only seen in the peptide cutter, just a cutter. Okay, we have to find out the peptide mass. We can only use the peptide mass. Okay, so next third one we go for PepDra. This is it make her. So next is one is translate. So this translate tool will come on the directly in the Google search tool also. So it is translate. That is nucleotide sequence will be converted into protein sequence. So we have already only having a protein sequence. So I take a nucleotide sequence from NCBI. This small size of so we can take this base post, copy and paste it in the translate tool. Okay, so in this we have find out sequences may be in a form of forward strand or reverse strand we have to convert. So depending upon the need, we can be able to use forward or reverse times. Forward or reverse 5 days to 3 days or 3 days to 5 days. Okay. So next we have to translate. So the results will be there. So open reading frame, that's the starting position for protein will be synthesis, that is in a rate to synthesis. So we can be able to use helpful for the initiator of one of the frames for iterating the amino acid sequences. So these are the translation. So we have to only pay the nucleotide sequence. So here that is AGCT88, that is indicates a nucleotide sequence that will be converted into protein sequence. So this is using the tool Translator. So next we use the tool Radar. So Radar is there. So we can okay. So Radar is not a Expasy tool. It is EMBA, that is European Bioinformatics Institution tool. So we have go for the EBI, AU, AC.UK services. Here we can type Radar, then you have to search, you will show the Radar. What does Radar means? Rapid automatic detection and alignment of repeats in the protein sequences. So here we have to paste the protein sequence. So once again repeat how we got to search, we have to open the Radar tool means this Radar tool. The Radar means it's a rapid automatic detection and alignment of repeats. Radar means is a rapid automatic detection and alignment of repeats. So this tool is we can search in EBI website. So here click the Radar, then you paste the sequence in the Radar page. Tool page we can find out the repeated sequences, by which number of repeats, where the place, position, the repeat of the sequences will be seen in this result. So this is the Radar. So we can able to download and take a printout also. So next one, this is the one of the Expasy tools. Here already I told we can search in the whole search bar. Likewise, we have interesting Expasy proteomic tools. So these are the different type of tools. What is the function of the tool is explained in this place. So PepMap means the mass of fingerprinting tool. Then PepC, this also a peptide mapping protein sequencing. So these are the more or similar tools. This tool will be that is developed by the different countries. So likewise, we can able to identification, characterization. We can use this tools. So this is Cleco mod means we can able to predict the oligosaccharide structures on the proteins. In the protein, you can able to find out where maybe a sugar glycoproteins are present. That will be found out the glycomod. Click and MOS means, so here it is, we can find out the oligosaccharide molecular weight. So next is the DNA to protein, that is translation. Translate, we can able to translate the nucleotide sequence to a protein sequence. Likewise, we can use this. Next is the similarity search. We can directly through NCBR, we can use the BLAST, or otherwise we can using EMBL also. There is a tool available for the similarity search, BLAST and FASTA. So here also there, EMBA is a FASTA 3 is an EMBA. So likewise, if you are interested, you can able to click every one of the tool and paste the sequence. What are the different type of results we have seen? Then now and then a tool that will become a best bioinformatic tool. So next the pattern and profile sources. Then it is a post-translation modification, topology prediction, that is a temp prediction, that is transmembrane region, where in which places this transmembrane will be present, that we can see through the template. Prediction of transmembrane regions and protein orientation. So how the protein will be present in the sequence. That is, we already seen in the intrinsic protein, extrinsic proteins, just like that. Sorry, there will be some network problem. There is some problem in the network, so it can't open. So we will try. Then it's a protoform. Then we can share about this one. Okay. So protoform. Here it is also present the radar. So we can click it. It will directly to the EMB website. So here it is also we have paste and then submit. Similar, already we see the result will be seen here. The picture sequence is very pleasant. So next, likewise, Statline is supporting sequence. So you have to choose every tool, click and open and paste the sequence and analyze the result. You will become a proteomic tool expert. So next is the second is structure prediction. So mostly we choose SOFAMA. So it is the bracket that given is the scientist could develop this tools. So another one, CAR 4, CAR 2, CAR 1. So they are the Indian scientists developed one. So these are the case sequence, submit. Send this name. We publish the program in the United Process Watch. There is an import problem very slowly. It's 1140 amino acids, so it will take more time. Okay, the result will be, see here. So first one is our protein sequence. Each amino acid is in each form of secondary structure. Each form of secondary structure. Then check it our length of amino acids protein sequence 1140 is correct. So these are the different types of secondary structure: alpha helix, theta helix, pi helix, beta bridge, extended strand, beta turn, bent region, random coil. So in this, which secondary structure is more predominant means alpha helix. Percentage is 13.3% of amino acids are involved in the alpha helix. And then next one, that is extended strand. This is random coil is more predominant. It is 71.14%. Next one, the alpha helix. So the more predominant secondary structure of this protein sequence is random coil. Second one is alpha helix. Third one is extended strand. Fourth one is beta strand. So these four different type of secondary structures present in this protein sequence. So question may be which one is the more predominant secondary structure present in the sequence, or low amount of or minimum which secondary structures are present in predominant or not predominant. So we can able to say that is it is which one is this is the random is the more predominant one, and low is 65 beta turn will be the less predominantly present. So this is about the secondary structure prediction. So we can able to go for another one type of GAR. This is the latest one. So here we have to paste the sequence. So here also the sequence is there. Here also the random is more predominant. The second one is alpha helix, and third one is extended strand. So what we can say about this GAR food? It is a secondary structure method version of Cornier, Hilbert, Robinson. They developed this tool. They published the paper in Methods in Enzymology. Okay. So these are the different type of tools available in Expasy. We can use the tool. We can able to clearly understand the our interest of protein interest and the structure will be we can easily predicted. So I wish you use these all tools and to become a expert in proteomics. Thank you.